Hello, my name is Caroline Schemmel, and I will be presenting today on severe acute respiratory syndrome, also known as SARS. SARS is a viral respiratory disease. Its causative agent is a coronavirus called SARS-associated coronavirus. Coronaviruses are a class of viruses named for the crown-like spikes on their surface. A corona is a crown in Latin, and the virus's name is directly derived from this word. And on this page, and you can see a picture of a coronavirus right here on the left. Specifically, SARS-CoV is a member of the family Coronaviridae within the order Nidovirales. SARS was discovered recently, in 2003, when an outbreak of the disease in southern China spread into a pandemic. And a lot of people have said that this pandemic, nothing like it has really been seen since the plague. So since the emergence of SARS-CoV in 2003, the number of known coronaviruses has increased from 12 to 36. As previously noted, SARS-CoV is a member of the family Coronaviridae and the order Nidovirales. SARS-CoV has undergone significant phylogenetic divergence from other known coronaviruses, and it is grouped with bat SARS-CoV as group 2b of the coronavirus family. The known human coronaviruses consist of human coronavirus 229E, human coronavirus OC43, and a variety of others, including the Middle East Respiratory Syndrome coronavirus. There are 6 different strains of coronaviruses that infect humans. Some of these strains cause the common cold, others pneumonia. There are also a variety of animal coronaviruses, including ones that target chickens, pigs, bovines, cats, and other animals. And SARS-CoV was discovered in 2003 and is believed to be of zoonotic origin. Like I said before, SARS is a virus. This is its structure. A helical nucleocapsid around the virus is formed by message-sense RNA, and a viral nucleocapsid phosphoprotein. Again, the large eye-catching and characteristic spikes in the virus envelope allow for the identification of coronaviruses via electron microscopy. And the spikes are composed of the oligomers of the spike S-glycoprotein. Their purpose is to bind receptors on host cells and fuse the membranes of the host with the viral envelope. SARS was discovered, as I said, very recently, only in 2003, and it was reported at that time because there was a pandemic of SARS. It was first cited in Asia, specifically in the Guangdong province of China, but over the course of the next several months, it spread to more than 24 countries in North America, South America, Europe, and Asia. During the 2003 outbreak of SARS, the World Health Organization reported that a total of 8,098 people around the world were infected with SARS, and of these people, 774 died. The United States had only 8 people with laboratory-confirmed cases of SARS, but all these people had traveled to parts of the world where SARS was rapidly spreading. And even after the end of the major SARS pandemic, another interspecies jumping event occurred in 2004. This event caused 4 more cases of SARS, but these are believed to be due to independent interspecies transmission events rather than being residual cases from the 2003 pandemic. And there were also 3 laboratory-associated outbreaks in Singapore, Taiwan, and Beijing in 2004, but these cases were very quickly contained. And the cause of the pandemic in 2003, it's thought to be of animal origin, that the virus jumped from animals to people. The transmission of SARS. SARS spreads largely through person-to-person contact, and this is believed to most often occur through respiratory droplets, which are dispersed into the air when an infected person sneezes or coughs. This type of transmission is called droplet spread, and it is characterized by droplets from the infected person being propelled a short distance, on average about 3 feet into the air, and then landing in the mucous membranes, especially the eyes, nose, and mouth of other people in the vicinity. Another means of transmission occurs when a person touches a surface that has been contaminated with droplets containing SARS, and then touches his nose or mouth. And concerningly, there's been some speculation that SARS might also be able to be spread through airborne transmission, but beyond the 3 feet radius, but this still has yet to be studied a little bit more. Transmission on average occurs on or after the 5th day post-exposure. During the 2003 pandemic, about half of the SARS cases were believed to have been acquired in healthcare facilities, and this is particularly due to treatment of SARS patients with nebulizers, suction intubation, and cardiopulmonary resuscitation, all of which generate and disperse a lot of droplets. So a lot of danger there. Some of the symptoms of SARS. Infection with SARS characteristically results in viral pneumonia with rapid respiratory deterioration. People infected with the disease first get a high fever with a temperature greater than 100.4 degrees Fahrenheit. They also get chills and general malaise. 10 to 20% of patients suffer from diarrhea within 2 days of infection, and a dry cough usually comes after 2 to 7 days, as well as the pneumonia. After about a week of infection, the SARS patient typically begins to majorly deteriorate. Their diarrhea becomes more watery, and chest radiographs show glass opacities and focal consolidations. Approximately 10% of SARS patients do not survive. The diagnosis and treatment of SARS. Unfortunately, there are no outward signs or symptoms of SARS that allows it to be differentiated from other cases of pneumonia. Therefore, laboratory confirmation is needed to achieve any certainty in the diagnosis. However, the type of laboratory tests usually done require type 3 laboratory security, and which most hospitals don't have. So a lot of work has been done to make a test that more hospitals could do in-house. However, because the pandemic in 2003 was so short-lasting and easily contained, not too much development has been made towards this project. As for the treatment of SARS, there are presently no approved drugs that are effective against any of the coronaviruses, including SARS specifically. However, there are several types of drugs being studied to target the virus. These include protease inhibitors, which might prevent the processing of RNA polymerase or cleavage of the S-glycoprotein, inhibitors of coronavirus acetylesterase, which might limit viral replication, inhibitors of membrane fusion, which would block viral entry, and also antibodies against the S-glycoprotein, which would also block the entry of the virus. Because no effective drugs specifically against SARS exist yet, SARS care usually consists largely of supportive care. And broad-spectrum antibiotics are used for this, antiviral agents, immunomodulatory therapy, that whole type of thing. Also, strict sanitation is really necessary when caring for SARS patients because it does spread so easily. Particular care must be taken involving the clothes that the people with SARS wear, because oftentimes the virus will become lodged in these things and can infect someone else. And it is advised by the CDC that anyone caring for a SARS patient have their temperature checked at least once a day to make sure that they don't have a fever and haven't contracted the disease. As you can see, there's a lot of parallels between this and the movie Contagion, or Pandemic. The future outlook of SARS, it's really actually amazing how quickly the medical and scientific community was able to identify and contain the novel SARS-CoV virus following the 2003 pandemic. Understanding the disease has really increased exponentially, and there are over 4,000 publications pertaining to SARS available online alone. However, many things are still not known about the virus, particularly as regards the mechanisms of its transmission as well as its pathogenesis. These gaps in knowledge mean that no effective treatment specifically against SARS or a vaccine exist yet. Research in these areas is still ongoing, as well as the development of better tests to help diagnose SARS. Concerningly, SARS and coronaviruses generally have an amazing ability to mutate via genetic recombination, and this could potentially lead to an outbreak like the one in 2003. The CDC thinks this is unlikely. However, the presence of SARS-CoV in animals, particularly their reservoir in horseshoe bats, keeps the possibility of another outbreak of the disease open. So the need for preparedness in keeping an eye out for SARS should not be ignored. Thank you for listening to my presentation, and here are my references.